Hi everyone, this is Dr. Rossler. So we're going to go ahead and get started on the week 8 lab. So we're doing this remotely for those of you who are in the Monday lab group, just because we were going to have to make them up on Fridays, on this Friday, and so we didn't want to do that. And especially for those of you who have a there's some people who have Friday evening labs, and that are Monday evening labs, and that would have been a Friday evening lab, so that would have really stunk. And this is actually a pretty straightforward lab of all the labs that we do. There is one part that is not good for doing it online, and so we're going to be making sure that you guys learn how to use the microscopes. That's the part in week 9 or week 10. We actually have time in one of those weeks. So we're going to make sure we get to that sort of active learning microscopy work then, okay? So don't feel like we will teach you how to use microscopes properly in case you don't know how. All right, so let's just go ahead and get to it. So let's look at, so just a little bit. So we're going to be using slides today that have been prepared for us, and we're going to be using a microscope to look at them. Just so you know, this is one of the things we would have talked about. Whenever you're carrying a microscope, it's really important that you use 2 hands, one under the bottom and one in the neck. All right, so I will make sure you do that and make sure you use it properly. So when you're using a microscope, and you may know this already, but there are 2 lenses typically for most microscopes, not all, but most microscopes have 2 sets of lenses, and the lenses, of course, in microscopes magnify. And so let's see, magnification, let's see, I think, yeah, magnification lenses are looking like this. All right, so this is what the shape of the lens is. I think it's concave is what that's called. Magnification, all right? And so there's 2 sets of these sorts of biconvex or bicon lenses that is going to ultimately magnify the slide that we have down here. So we're going to ultimately be putting a slide down on the stage. That's what this is called, a stage. All right. And we're going to be putting glass slides into these little brackets there so that we can actually look at our objects that are on the microscope. All right, so we'll be sure to teach you how to do this. Now, one thing just real quickly, there's, again, like I said before, there are 2 series of magnifications that occur. And the top magnification up here, the ocular lens, let me make this red. This ocular lens always or typically has a 10x magnification. So that means that when you look just through the ocular lens, you're seeing 10 times better than the eye sees. Down here, you've got the specimen lenses, and there's usually 3 or 4 of those. Here you can see the different types of, on the next slide, we'll be able to see all the different lenses that we use. The first and the lowest one is a 4x lens. And so the 4x lens is obviously 4 times better by itself. But what happens when you use it in combination or in tandem with the ocular lens, so there's a 10x lens on top and a 4x lens on the bottom, it actually ends up magnifying 40 times better. So you just multiply those 2 values together, and that's actually how you see. So whenever you're looking through a microscope or telescopes, they have these different magnifications. And you would actually, if they're in tandem with one another, you just end up multiplying each individual magnification. All right, so usually when you use a microscope, it's always best to start off with the lowest one first, and then gradually get higher and higher in magnification. So if we look at the next slide, go to that. We're going to see again the different types of magnification that we can use. And as we said before, the ocular lens is always tenfold. And then we'll usually start off with the scanning microscope, which uses a 4x lens. Oops, went too far. And again, like we said before, that would have a total magnification of 40. And then if we want to go into more detail, we'll go up a stage. We go up gradually rather than going straight to the end. The next step up is usually a 10x lens. So 10x times 10x is obviously 100. And then for a lot of the things that we're going to be working on, we're not going to go any higher than lower power or high power. High power, of course, here you can see is 40x, so that would make a total magnification of 400. Every once in a blue moon, especially when you guys get into microbiology, you're going to use an oil immersion lens, which is really quite fun. And it's a 100x lens in the objective lens. That coupled with the ocular lens 10x, this course can give you 1,000-fold magnification. So yeah, there's they're pretty fun to use. And so we're going to be using these microscopes today. to essentially magnify images. Well, you guys are not going to be doing the steps. We're going to be looking at those later on. So I'm just going to kind of go on to the part of the lab that is going to ultimately deal with mitosis, which is the second part. All right, so mitosis. So again, for today, your lab report, you're going to talk just a little bit about the theory behind microscopy. And we've talked a little bit about that there's, again, different lenses of different magnification strengths. And we're going to be able to use, again, a combination of those to be able to look at our object on a stage. The object that we're going to be looking at today are onions, right? So we're going to be looking at onions that are the roots of the onion that's actually actively undergoing growth. So when an onion is growing and sending out shoots, obviously those shoots are mitotic cells, and we're going to be able to look at those onion roots and as they're actively undergoing mitosis and determine at what stages they are. All right, before we go into the stages, you may have heard a little of this in high school. When we talk about cells, right, cells have to often divide. And that process of division is called cell division, and more specifically mitosis. So the mitosis process is ridiculously important. It kind of explains how you started off as a one single-celled organism, right? When your mother and your father created you via conception, right? You started off as a single zygotic cell that had been an egg that had been fertilized by a sperm and became a zygote. And then that one cell grew and divided and divided and divided and ultimately became the 2 trillion cells that makes up you, right? So that's kind of amazing. Almost all the cells in your body are identical to each other, and that's a really important part. They all have the same genetic information for the most part, and we want to copy that DNA very faithfully every time we make a copy of a cell. Okay, so that's the big purpose of the cell cycle. Big parts of the cell cycle are kind of involved in regulation of that division and growth, right? So let's think about where we start. So here would be the starting point. So think about it, you're a brand new cell. So here's where it would begin. So that brand new cell has just been created. It is going to exist for a very long period of time. Usually it can exist for up to about 20 hours or so in this interphase period. So this whole area over here to here, most of the cell's life is spent actually in interphase. And then interphase, as you guys probably know, it's made up of 3 steps. The first step is G1, and that's really where the cell is just kind of hanging out, right? It's just doing its normal, jobs and whatnot. And then at some point, depending upon the environment, so there's various environmental signals that will be received by the cell. Some of those environmental signals are like growth hormones, or we could think about when you get a cut, right? If your epidermal cells are not touching another epidermal cell, they're not happy. And so again, the lack of contact with an adjacent cell would send them into mitosis. Going through the cell cycle. So there's a lot of different things from the environment, environmental signals the cell will receive. And again, if it receives enough of those sort of signals, again, growth hormones are a great example of this, those signals then are going to convert that G1 cell into an S cell. Now, to do that is a lot of work, right? The cell is going to check. It's like we like to think of it as a stoplight, right? So before the cell just willy-nilly goes into the S phase from the G1 phase, it's trying to make sure that everything is just kind of just so-so, like this really needs to actually happen, right? Because cells don't want to waste all this energy to divide unless they're really needed. All right. And we know what can happen, of course, is that in some situations, the cell will divide when it's not supposed to, right? And if that cell divides when it's not supposed to, you're going to get a tumor, right? And tumors, of course, are not necessarily that good, typically bad, and it, of course, is a hallmark of cancer. Not all the time, but oftentimes, right? All right, so if that cell does receive enough of the signals and gets the green light to go into division, right, what's going to happen is that G1 cell is going to go into the S phase. Now, what happens in the S phase is that the DNA is duplicated. And so let's just talk about what we mean by that. Your DNA in the G1 phase, right, think about you are a diploid organism. Diploid organism. So that means you got 2 copies of every chromosome. You got one from, say, your mom, and you got one from your dad. That's a mother and a father sign there. And this is referred to as a homologous pair, right? So let's think about, say this is chromosome number 5. All right, chromosome number, you got a lot of chromosomes, right? You got 23 pairs of chromosomes. And let's imagine we're just looking at chromosome number 5, right? And so one of those chromosomes, 5, is from your mom, and say it contains the gene for eye color. All right, and your dad also gave you a chromosome 5, right? And it also contained a gene for eye color. So that's why we kind of talk about genes oftentimes in pairs, is because again, you got one from mom, one from dad. And again, they're most likely not going to be completely identical to each other, right? We talked in the last lab about polymorphisms, like there's these little subtle variations in DNA sequences between organisms, right? And so your parents, again, different people, right? You might have, say, a big B from your mom and a little b, meaning a dominant and a recessive allele from mom versus dad, right? So they're subtly different from each other. They could be the same. You might have both gotten, you know, a little b or a big B from your mom and your dad, but again, there's tiny little differences. Long story short, is that in G1, you are a diploid creature. You got one copy of every gene type or chromosomal type from mom, one copy of every chromosomal type from dad. And that's why you're quote unquote diploid, 2N. Now, what happened at the S phase though, right? If you received enough signals and it went into the S phase, is that each of those homologous pairs is gonna duplicate. So my mom's chromosome duplicates perfectly, God willing, right? And my dad's chromosome copies itself perfectly. So now I've got 2 copies of little b for my dad and 2 copies of that, say, big B for my mom. So this is happening at the S phase. This is a lot of freaking work. Let's think about it some more, bedazzle you. There are 6 billion nucleotides in your nuclear genome at the G1 phase. So you've obviously copied all of those nucleotides. So you've gone to 12 billion nucleotides. That was a lot of work for the cell, right? So that's an incredible amount of effort. And the cell only wants to do that, like we said before, when it gets the green light and that all systems are go. Now, a lot of cancer drugs, as you can imagine, are going to target this site. So if you have a cancer cell, what's happening is the cell is always going from G1 to S to G2 all the way around very, very, very quickly, right? And it's causing the cell to grow erroneously. Just to show you this, actually, I've got some fun stuff to show you. Let me see. I was quite tickled. Oh, have you not been seeing this? Oh, damn. Let me pause this for a second.